Okay. Good afternoon, everyone. Thanks for attending to our new Sussex Vision talk. Today, I have the pleasure of introducing our next speaker, Professor Henrique von Gersdorff from the Volume Institute. Thanks, Henrique, for accepting our invitation. It is a pleasure to have the opportunity of enjoying your research, your recent research field. And briefly, I would like to give a brief introduction about Professor Henrique von Gersdorff. Professor Henrique von Gersdorff got his PhD on physics at the University of Minnesota in the United States. Then in the field of the neurobiology, he did two postdocs, firstly with Gary Matthews, and then he moved to Germany at the laboratory of Professor Erwin Neher in Göttingen for studying some really interesting mechanism of vesicular plurifilins, if I am okay with this. I remember that during my PhD, I read many of your papers, so you are cited in many times on my thesis. So, and today, Professor Henrique, so actually, he's working at the Volume Institute in the United States, and he is working as a full professor and senior scientist. So, thanks, Henrique, again. So we are ready for enjoying your talk, and thanks for accepting our invitation again.
Thank you a lot, Jose. It's a pleasure to be here. I've enjoyed a lot of these series of lectures, the Sussex Vision series. It's been wonderful to see the community. So I think it's one of the good things out of the pandemic that we found ways to still connect with each other. So thank you very much for the invitation. I hope you will enjoy the talk. And let me start. I will hit my screen share here. Can you see this? Yeah. Good. Yep. Okay. So, so this is the title of my talk. And I'm going to be talking about crossover inhibition in the mouse retina, which is a feature associated with the A2 amacrine cells. And this talk will also be concentrated on the glycine release from these A2 amacrine cells, these specialized amacrine cells, which are narrow field glycine releasing amacrine cells in the mammalian retina. The work is by Mark Meadows, Vera, he's a PhD student in our lab, and Vera Balakrishnan and Xiaohan Wang contributed at initial stages of the work, especially the capacitance measurements. And so let me start out by showing you some pictures of what the mouse retina, since we're talking about the mouse retina, has to worry about. And one of the things it has to worry about is these creatures here. This is the accipiter shark-shinned hawk. And it likes to hunt actually at dusk and dawn when maybe there's less contrast around. And in order for the mouse to detect this creature, you can already tell that it would be good if it had retinal ganglion cells that would be orientation selective, so that it would be able to tell the orientation here of these two bars, let's say, dark bars. So orientation tuned retinal ganglion cells would be a good thing for it to have in its retina. Also directional selective, right? It wants to know if this bird is flying in this direction or that direction. And it also would like to tell the velocity. So the velocity at which the bird is flying in the air. And it has UVs, so there are UV cones that allow it to see well in the UV range. Here we have a lot of UV light during the day. And the other feature, of course, that it wants to see is this object getting smaller, which is a good thing, or is this object actually getting larger quickly, which is a bad thing for the mouse. So it wants to also detect whether this object is looming or not. And it has to do that, you know, in dusk and dawn when it starts to get hungry, and it's a nocturnal animal, the mouse, and it wants to start looking for food. It has to be having good vision. I think mouse actually have good vision. They cannot detect this just with their noses or with their whiskers or their ears. These are silent flying machines. You can see the aerodynamics of this. So the mouse probably has very good vision, a very sophisticated retina to detect these objects. And these are really formidable creatures. You see the size of the eye is huge. You can think of it compared to the brain. And if you actually dissect this out, so I found this in the web, you can actually think of these accipiter hawks as being flying, primarily a flying set of eyeballs. You see these huge eyeballs compared to the brain. So it probably has a lot of space here, neural space dedicated to vision, to be able to scan and then swoop in and dive for its ability to survive through hunting. So contrast is important, and contrast in low light conditions, dawn and dusk, when the mouse really needs to see these creatures if it wants to venture outside. So what are mechanisms that the retina has for contrast? This paper I wanted to quickly review. So in my initial part of my talk, I will review a few papers that I think are really interesting for this concept of cross-inhibition, which is how the on and off pathways of the retina can talk to each other. And this paper is really, really nice from Michael Fried lab, talking about the efficiency of contrast encoding in the mammalian retina using this crossover inhibition. So if we think of the cones and how during dark, so if you give a dark flash here as your stimulus, the cone will release glutamate to darkness. It will activate these AMPA-kainate receptors here in the dendrites of the off bipolar cells, which will then release glutamate and cause an EPSC, a depolarization in these off alpha cells, these off ganglion cells, and that will cause spikes. So communicating to the brain that there's a flash of darkness in the field of view. At the same time, you will have also an inhibition, so there will be glutamate onto these on bipolar cells, which will be hyperpolarized. They're connected, these on bipolar cells, this on pathway here through gap junctions to these A2 amacrine cells, which is the main interest of my talk will be to try to understand a little bit better how these cells function. And these cells then receive that that hyperpolarization from coming from the signaling from the cone, and that will inhibit these the glycine release here at the terminals of the A2 amacrine cells, so that you'll get even more efficient release of the glutamate. And you might think that this, and actually the release of the glycine occurs not only here presynaptically at the terminal of the bipolar cell, but also onto the dendrites of the off alpha cell. Actually, even the soma recently, Will Grimes and Jeff Diamond show that even the soma gets direct glycine release from these A2s. So they're very versatile cells. And if you now block this pathway with a drug called LAP4, what happens is very interesting. So if you don't have this crossover inhibition, here is some data that they show in the paper. So in control, you have a very low noise. So you have here a little bit of contrast, 4% contrast, and you get a depolarizing current here. And here's another reproduction of this. It's very highly reproducible, low noise, very good signal. You put LAP4, it starts to get very noisy, the baseline. And it becomes very difficult for you to distinguish what is signal from noise. And if you do an analysis of the noise and the signal here in control, you can distinguish it very well with these Fisher measures. But in LAP4, you cannot distinguish anymore these low contrasts. So this circuitry here is vital for you to detect these low contrast features. And this is all in voltage clamp. We have currents here. If we go to current clamp here and look at spiking, we see that the important features for spiking are, of course, the resting memory potential of the off alpha ganglion cell, and also how distant that is to the threshold. So here's the resting memory potential, the plateau potential, where you start to get spikes, you get a burst of spikes to signal to the brain. And where you have your threshold is very important also. And as you depolarize more, you get more of this plateau potential, you'll get more spikes. And you see that in control, you can be safely below the threshold for getting spikes. So you might not be able to detect very, very low contrast, but if the contrast gets a little bit better, you get a burst of spikes. Whereas in LAP4, you get very noisy, you get spiking all the time because the cells now don't have that tonic inhibition. The resting memory potential is more depolarized, and now any little glitch here of the memory potential will reach threshold, and you get a lot of spikes. You might be able to get even some more contrast some more signaling to very low contrast, but there's too much noise for you to really distinguish that. So, so you can see how important this A2 amacrine cell is, because it sets the resting membrane potential at a sweet spot where it's not too hyperpolarized, where it's far away from the threshold. It's not depolarized, where it's giving lots of spikes. It's really setting very delicately this membrane potential so that you get it very finely tuned to get very efficient contrast. So you can see how this mechanism of crossover inhibition can be very important for detecting these dark flashes. Another situation, I was telling you about the hawk and the mouse, and how if the hawk is looming, the mouse wants to very quickly detect that, send that signal to its from its retina as quickly as possible to its motor cortex so that it goes back to its burrow very quickly. So it doesn't want to have looming detectors or orientation bars in its V1. It wants to have it in its retina to get the message very quickly to the brain. And this group found this really interesting lumen detector. And interestingly, the A2 is critical here too in the circuitry. And they say that the circuit's essential building block is rapid inhibitory pathway that comes in part from the A2. So they studied these PV5 ganglion cells that actually do not respond to lateral motion, but they do respond to looming motion. So an object that's small and gradually gets bigger and bigger and bigger, they respond to that very, very well with spiking. And in this paper, they even did paired recordings between the A2 and these P5 ganglion cells. And it's really interesting. They could get pairs. Here they show how these iPSCs are are blocked by strychnine. And here's the A2 that they reconstructed. Here's the ganglion cell, the PV5. You see it has this large dendritic field. Here's one A2, which are these narrow glycinergic amacrine cells. That's synapsing. They show here, these are the dendrites of the PV5. There's synapsing, and here are the lobules that release the glycine onto the dendrites of these PV5s. And so for looming detectors, maybe the A2s are really a critical fast glycine inhibition that's important for these cells to distinguish from something that's moving laterally from something that's moving. So really nice circuitry here for a feature detector. The last thing I wanted to say, sort of as a general overview of why A2s are so interesting, they're actually the highest density of amacrine cells that we know is the A2, so they're very important. This paper I found really fascinating from Erika Stretoy and Rania Masri and Ulrike Gruner, who's in Australia. And they looked and saw that there are A2 amacrine cells in the primate fovea. And this is actually from human retina. Here's the figure caption. And what they're doing here is, this is just a beautiful picture. They're putting rod arrestin to look at where the rods are. And you see here's the fovea, the foveal pit. And you see how the rods are petering out here. They're petering out. And you have only the cones and cone bipolar cells underneath here. They also image the rod bipolar cells. So you see that the rod bipolar cells are petering out here. And they looked at calretinin as a marker for the A2s. It turns out that it's a very, very good marker for A2 amacrine cells in primate retina and human. And here they even reconstructed, in this rectangle here is shown blown up here, and you see reconstruction of these three little midget amacrine cells, which are really much going quite into the fovea. So pretty fascinating. They even in this paper speculate that A2s are probably very ancient amacrine cells that developed together with the cones. And only later in evolution, when the rods came in, because mammals like rats and mice, they figured out that hawks sleep during the night. So they figured out we better become nocturnal so we don't have to face those hawks. And so they started to develop rods for night vision, and co-opted probably the circuitry of the A2 so that it would serve the signaling of single photons, for example, the low noise pathway for single photons. So the A2s are pretty amazing. They're involved in the low noise single photon circuitry, but also in the daytime, as this shows very nicely. So A2s are ancient cells that probably co-evolved with the cones for this crossover inhibition. They're multitaskers, as I was showing you. They are involved in detecting lots of features, not only in the circuitry to count photons, where it's scotopic, but also in the photopic range. And so they have a rich, let's say, social life, because they're connecting, receiving inputs from rod bipolar cells and cone bipolar cells. And they're also hooking up with many different types of bipolar cells, sending, gap junctions, electrical synapses, chemical synapses to different bipolar cells, and also releasing glycine to retinal ganglion cells and setting their resting memory potential in a very finely tuned way. But the question I want to ask today is, okay, they have this nice social life, but what about their interior life? And when I say interior life, I don't mean that in sort of, a spiritual way. I mean it more in a biophysical mechanisms way. So what are the calcium stores and intracellular messengers? if these cells are such good multitaskers, they probably have a rich internal life of lots of different second messengers and G protein coupled receptors and intracellular messengers like cyclic AMP and phosphate and calcium to signal. And actually, we don't know too much about how second messengers are modulating the release of glycine. And that's what I want to tell you about, how our lab is trying to approach that and see how this rich interior life can facilitate and promote the multitasking of the A2s. Okay, so this is just a review here of the retina. It has these multiple parallel pathways, lots of different types of bipolar cells. And I want to focus on this glycine release that's very important for crossover inhibition, so that these excitatory signals coming in here are converted in the A2 amacrine cell to inhibition in the off pathway. And so these A2s are central to both scotopic and photopic vision. We used to think, when I was starting graduate school, that it was just specialized for scotopic vision. But then we started to see was this disinhibition and contrast enhancement and the looming detector, that the A2 not only has, a night shift, as Jeff Diamond has famously said together with Nick Oish, but also has a daytime job, a daytime gig, where it helps to detect all these features during the day. So we're going to look at these dendritic inhibitory release sites at the lobular appendages. So these are called lobular appendages over here, these little bubbles connected by necks here on the amacrine cell coming out from this thick apical dendrite. And they also have gap junctions. So quite complex, but very small cells doing a lot of different tasks in the retina. So pretty interesting. And the way we've looked at the glycine releases was use this technique where we can look at living cells and measure their membrane capacitance to look at the recycling of synaptic vesicles, the exoendocytosis of synaptic vesicles in synaptic terminals. And I learned the technique from Gary Matthews when I was a postdoc in his lab. And he had this wonderful preparation, which was the goldfish giant mixed bipolar cell terminals. And so here you see the goldfish bipolar cell, very similar cells, but much smaller in the zebra fish retina. And you can actually acutely dissociate these cells from the retina. They often come with their axon cut. So you have this perfect sphere that's ideal to measure membrane capacitance, which is a measure of the surface area of the cell. So we would measure the capacitance and then depolarize it, elicit an L-type calcium current, get calcium influx, which triggers the exocytosis of vesicles, which triggers the increase in the surface area or the capacitance jump that you see here. And then the vesicles, after they fuse and increase that surface area, we found that they were re-internalized. So the capacitance returned quickly back to baseline. And we interpreted this return as being endocytosis. And so here is just a summary of the work that Gary Matthews has done. This all comes from the lab of Gary Matthews, that there was very fast endocytosis. Endocytosis occurs immediately. Within 100 milliseconds, you already start to see a decay of the capacitance. So very, very fast, so-called ultra-fast endocytosis was first found in these nerve terminals. We also went on to study, by changing the pulse duration, the release of the nerve terminals. And we saw that there was a very fast phase. So if you take the first derivative of the capacitance, you see that there's this spike, a very fast release phase, followed by a slower, more delayed or tonic release. And this turns out to look very much like what the glutamate that's being released from the nerve terminals looks like. There's a fast component and a more delayed, sustained component. different bipolar cells will have different kinetics for different temporal frequencies that they're encoding. Some will be very fast and transient, and others will be more sustained. And this all comes from the work of Gary Matthews. He was just a wonderful person and a wonderful mentor for me. And when I was transitioning from physics, he really taught me how to become a biophysicist, even more so a biologist, to think like a biologist in a quantitative way, and had a lot of contributions to the retina and synaptic transmission. Sorely missed, just passed away way too young. But we used then this technique by recording from the somas of the A2 amacrine cells. And our hope was that since these necks here are very short and these ends here are sealed, they're small little bubbles, they're short here, you see them here, that this would be electrotonically quite tight, at least this part here, and that we could still use the capacitance. So although this is by no means a sphere, which is ideal for capacitance, we could still clamp this area here very, very well. And so we just tried to see if we could transport this technique of capacitance measurements to these A2 amacrine cells. And in fact, it worked quite well. If you just hold the cells at minus 80 millivolts and depolarize, you see, and you put a sine wave here of 2 kilohertz, you see that that depolarization elicits a calcium current. These cells have a single type of calcium current. In that way, they're simple. It's KV 1.3 non-inactivating calcium currents. This calcium influx then causes excitation of glycinergic vesicles, we hypothesize, and that causes this jump in the capacitance. There is a change also in the series resistance. We typically see a small downward shift. This you would predict exactly also happens, for example, in the mossy fiber terminals. Stefan Hallerman has seen this in his capacitance measurements. These look topologically very much like a mossy fiber terminal, with the release sites very close to where the patch pipette is. So it seems like it's okay to measure capacitance. So we went ahead and did that and published already several years ago a paper on that. And you see that as you increase the pulse duration, you get bigger jumps, more exocytosis. The exocytosis doesn't wash out for the first 10, 15 minutes. It's pretty stable. And so you can now do the same kind of experiments that we were doing in the bipolar cell with Gary and see that there's a very fast initial release. So also very phasic release, and subsequent paired recordings by Cole Grandin and Jeff Diamond and Meg Faruqi and Espen Hartveit between the A2 and the cone bipolar have shown that they have a very fast component of glycine release, followed by a more sustained component of glycine release, very much like what this capacitance seems to suggest. Okay, so we can do capacitance. And how does the ultrastructure look like? Are there synaptic vesicles? And yes, this has been known. There was this beautiful paper from Stretoy, the same one who was working on the fovea I showed you before. She worked with Raviola and Deschaux in the early 90s, mid 90s, and had these beautiful electron micrographs. So the A2 amacrine cells have these beautiful active zones. They're packed with vesicles. Here you see 3 different active zones, even very beautiful clathrin-coated vesicles here, and even some clathrin right at the membrane. This is the cone bipolar cell terminal here, receiving the glycine. And here's another amacrine cell that's synapsing also here onto the cone bipolar cell terminal. So a lot of microcircuitry here processing the signal. This was ultrastructure from the rabbit A2 to off cone bipolar cell synapse. So multiple active zones, which is probably what's giving us that big capacitance jump. Actually, the capacitance jump was at 40 femtofarads, which is equivalent, if a single vesicle is 40 attofarads, to about 1,000 vesicles, quite a large number of vesicles. That's over the whole surface of the cell, of course. And so there's a large reserve pools because it just seems to not really deplete. If you depolarize not too strongly, you can keep on releasing. So maybe there's a recruitment process of vesicles here towards the active zone. And we asked questions about modulation of G proteins and dopamine and cyclic AMP. Dopamine seems to act through cyclic AMP. There's good evidence that glycine can be modulated by dopamine from the lab of Erica Eggers. And we're also interested in the stores. So what's the interior life of these cells like? So the first thing we looked at was cyclic AMP, and that's derived from ATP. So we did an experiment where we just simply put 1 millimolar cyclic AMP into the patch pipette, and we recorded the capacitance 1 minute, let the cyclic AMP dialyze fully into the cell. And when we did a second measurement, we saw a nice potentiation. And this potentiation does not happen if we don't put 1 millimolar cyclic AMP. And so here's the average data. We see a highly significant increase, a lot of variability. So one A2 to another has very different release, but on average, there was a highly significant increase whenever we had 1 millimolar cyclic AMP in the patch pipette. The calcium currents did not change significantly. So this is the total charge in the calcium currents. We then went on to look at the mechanism, and the mechanism is very interesting. It turns out that the cyclic AMP is not acting through pKa, which was our first guess. Actually, pKa acts at the gap junctions in the arboreal dendrites of the A2. But for the glycine release, it's this molecule called EPAC, which is not a kinase. It binds to cyclic AMP, has a large conformational change, and then can act on different targets. And so to show that it was EPAC2, we used a selective inhibitor. And with that inhibitor and 1 millimolar cyclic AMP, it completely blocked the potentiation without significantly changing the calcium currents. And whereas if we use the selective acyclic AMP analog, so something that selectively activates EPAC, we saw the nice potentiation still occur. So this was very good evidence that EPAC is involved in this story. And in our paper, which was published back in November in Journal of Neuroscience, we show that there's an increase in the vesicle pool size. And this does not, without any change in release probability. We did that with paired pulse experiments. We put a blocker called H89 of pKa. That does not block the inhibition, so we don't think pKa is involved. I'll show you some experiments where we use forskolin to directly activate adenylate cyclase to increase cyclic AMP in the cells. And that actually works beautifully. So more evidence that there's cyclic AMP through adenylate cyclase. And then I'll show you some evidence that there are internal stores involved here. So let's get at how we looked at forskolin. And so for that, we wanted to do a different set of experiments where we would patch the off bipolar cell and look at how glycine, spontaneous glycine miniature iPSCs are occurring just by patching these off bipolar cells. And we initially did it in rats because these off cone bipolar cells are very small, hard to record from, and in rat, they're bigger, so it's a little bit easier. We also did it in a transgenic mouse, a SIT2 transgenic mouse. I'll show you some of that just in a bit. And we can distinguish very well the off cones from the on cones, which are shown here. And here's our data. So beautiful signal to noise ratio. You see that before treatment with drugs, here's the noise, here's the signal, very nice minis occurring spontaneously, completely blocked by strychnine. And if we put forskolin, we get a nice potentiation of the frequency of the events. And the analysis is shown here. We had a large number of events, and you see this is the inter-event interval. So we're getting more short intervals, which means higher frequency of these events. And that's what's shown here. So here's the semi-log plot showing better these small intervals. And here's the frequency plot showing that on average, there was a significant increase in the frequency of the minis. It's about more than a 50-fold change. We also looked at the amplitude and the kinetics of these minis. They're very fast, surprisingly fast, so about 2 milliseconds to decay. Whether we had it in control or with forskolin didn't really change significantly. Here's the amplitudes, so nice skewed distributions. The amplitudes didn't change very much with forskolin. This is the transgenic animal we used. This was actually a trick we heard from Cole Grandin and Jeff Diamond. They use this in paired recordings. So it nicely labels the off-cone bipolar cell for a certain class of off-cone bipolar cells that receives this glycinergic input. Here's a typical experiment. One experiment we did where we put some forskolin, and you see there's a baseline rate of minis, a certain frequency of minis. Then after just a few minutes, it starts to rise and stays up. And so here's average data. You see after a few minutes here, 4 or 5 minutes, you start to get significant increases in the frequency, and it remains highly significant later on after 8 or 9 minutes. Okay, so this collaborates what we're seeing with the capacitance. Here, I just wanted to show you some nice images of that we patch these cells with the SIT2, a nice spinning cell. So I like this video because it's just beautiful how the cells have this intricate nerve terminal. And this is from Heinz Wessler's review. And so this is the type of off bipolar cell that we're looking at here that has this SIT2 expression. Okay. And then just for comparison, I find this interesting to compare these glycinergic currents in the terminals of the off-cone bipolar cells with the glycine. Remember, I was telling you that the glycine also hits the retinal ganglion cells, and in both the dendrites and also even the soma now from the recent work of Will Grimes and Jeff. And here the decay is much slower. Here you see the time constant is 20 milliseconds. So actually, if you were to fit these EPSCs, average them out, it's more like 20 milliseconds decay here. So very different kinetics. And I just love this picture from my old friend Dario Prati in the lab of Isabel Llano. We overlapped as postdocs when in Göttingen. And I was just amazed by these records where you see the decay of the EPSC and then single channel currents occurring here. And you can see that the distribution of these iPSCs in the retinal ganglion cells is quite nicely Gaussian, big amplitudes, and large conductance for these glycinergic ion channels. So interesting comparisons. Okay, so the next thing we wanted to look at was stores. We did some pharmacology. There's 2 drugs that we used. One is called 2-APB. We got this from a paper of Martin Wilson, where he did this also in the dendrites of cultured chicken amacrine cells. And he used this drug, and it's claimed to be an inhibitor of IP3. It's actually kind of dirty drug. It might be a better inhibitor of these SOCI, the store-operated calcium exchange channels, and it might be depleting the calcium stores. So it's disrupting the calcium stores. So we tried it, and in fact, it did block the potentiation with cyclic AMP. Here, we're leaving the 2-APB 10 minutes on also the ranitidine for 10 minutes, and then we're doing the whole cell patching, so letting it incubate into the slice. So we blocked the potentiation of cyclic AMP, but there was also a slight drop of the capacitance, the calcium current. Then we did ryanodine. We did 2 different concentrations of ryanodine. And here you see that there was a very significant, very nice block of the potentiation with ryanodine, with an overall very small change in the calcium current. Actually, if we use 10 micromolar, we saw no change in the calcium current and a complete block here of the potentiation. So this was encouraging. We think it collaborates very well. Previous findings from Martin Wilson and Chavez and Diamond also had nice papers with the rod bipolar cells showing that they also have calcium stores. But they were careful in their paper to say that this is not coming from the A2 amacrine cell. Some other glycinergic cell is actually doing this. Okay, so there seems to be stores. That's cool. If there's stores, then it would be very interesting to try an experiment where we put high GTA to see if we can block that effect. And so that's what we did here. We first did control experiments with just 2 EGTA, which is our standard. We see beautiful potentiation here. And then in the same slice, we changed the patch pipette and used 10 EGTA. This is now unpublished data that I'll be showing you next. And you see very nice block with 10 millimolar EGTA with still 1 millimolar of cyclic AMP in the patch pipette. So this collaborates very nicely, this idea that there are stores. You can block the calcium from the stores, which are probably a little bit distant from the active zones. Interestingly, very interestingly, the total capacitance jump didn't change when we went from 2 millimolar GTA to 10 millimolar GTA. So this seems to suggest that there are nanodomains in these cells. So one conclusion we can have is that these A2s have a tight nanodomain coupling between the calcium channels and the exocytosis calcium sensor, whatever that is, some type of synaptotagmin probably. And the release from the stores, the release of calcium from the stores is further away from prime vesicles because we can actually block it with 10 millimolar GTA. And so calcium stores may mobilize vesicles. Our idea is that in control situation, the cyclic AMP is releasing through EPAC this calcium from the stores, and that mobilizes vesicles that might be in the cytoplasm so that they move to the active zone and increase the releasable pool size. Okay, we would like to get some more evidence for these stores, more direct evidence. So we used a GlyT2 Cree mouse to try to express some GFP. So first, GlyT2 was a bit controversial, whether it's really expressed in the A2s. So we did a cross between this GlyT2 Cree and the Phlox TD tomato, and we saw beautiful fluorescence all over the retina. This is actually from Ben Sevier. Beautiful images that he obtained with a confocal. Here you see in the INL the cell bodies, and here you see these little lobular appendages in the IPL. So it really looks like the A2s. Here's a cross section. It's a bona fide A2 cell that this GlyT2 is expressing the TD tomato on. So then we crossed with GCAMP, the calcium sensor, genetically encoded calcium sensor. And now we can do experiments, for example, like this one, where we just patch the A2 and depolarize it and see what happens. And we see really nice signals in the lobules as soon as we depolarize the cell. So the lobules just light up and the soma also lights up. So let me stop this video. And so we did analysis of the different lobules, and you see very nice rise in fluorescence and then a decay. It's kind of cool to compare the kinetics. The lobules decay much more quickly than the soma. It's very similar to a nice study that Tom and Leon did on the terminals of the bipolar cells, and smaller terminals have very fast decay because of the surface to volume ratio being so favorable. And here we compared the fast time constant and the slow time constants, and the lobules have just a half second here time constant, whereas the somas have this slower time constant. So they decay more slowly. So there's the analysis of the imaging. Now we could do more interesting experiments, which is to put cyclic AMP. So there's this drug, 8-PCT, that's an analog of cyclic AMP that goes right through the membrane. So we can just puff did some quick and dirty experiments. We just puffed some of this drug onto the retina. And there you see it lighting up the A2, and you're going to see another two, they're flickering in here. And there you go. So it's like lightning bolts in the sky here, the flickering of these A2s whenever there's cyclic AMP releasing calcium from stores, we hypothesize. And here's just analysis. So we looked at these two regions here, and then after a few seconds, one A2 lit up, a few more seconds, another A2 lit up when we're perfusing this drug over the slice. And here we looked at different regions of interest. Here's the analysis of the data. So we put the 8-CPT, and here's little bursts of calcium coming out. So this is also unpublished and kind of preliminary data, which we want to analyze further. We repeated this here. Some signals are more sustained, some are faster. They occur quite quickly once we put the drug in. We also tried caffeine, same cells, so also saw caffeine. And this cell here, I think, had way too much caffeine and just petered out. It's probably zonked out on the caffeine and is not recovering. Okay, let me reach my conclusions then. I showed you that there is a rich interior life in these A2s, that they have adenylate cyclase that can convert ATP to cyclic AMP, probably through some G protein receptor. We're not sure what that is, maybe dopamine, activating this EPAC molecule, which activates these ryanodine, IP3 channels, releases calcium, promotes the recruitment of the vesicles to the active zone, where they lie within nanometers of calcium channels for fast release. Remember that looming detector was critical that inhibition be very, very fast, and the A2s provide very fast inhibition. So we think there's adenylate cyclase through EPAC and calcium stores. It's an interesting mechanism that increase the pool size. What are the unknowns? Well, we don't really know what the GPCR, the G protein coupled receptor signaling is. What is it dopamine? Is it some other neurotransmitter? We're working on that. We also think there could be maybe EPAC working on RIM and directly on the fusion machinery. There's evidence from insulin cells that might be happening. So we want to look at that. And then there's some interesting mobilization of vesicles, maybe through proteins like piccolo, to the active zone to get this pool size to be increased. So those are some of the questions we want to explore in the future. The main person who did this work was Mark Meadows, really talented, wonderful student that I've had. And here he is actually in Norway. He visited Meg Faruqi and Espen Hartveit in Norway. I think this is one of the tallest peaks there near Bergen. And these are our collaborators at OHSU, Ben Severe, Catherine Morgans, Anusha Mishra, Steve Smith, all the people in our lab. Also a shout out to Teresa Puzzeri and Roland Taylor, who helped us a lot in these A2 recordings. And Meg Faruqi, who hosted Mark. He learned a lot with them in a very short stay there in their lab. Our funding agencies, and here is Dusk in Oregon, and you can actually see hawks flying at that time of the day. And here's Mark in Berlin, and he's gotten a taste for beer And I think he's probably going to do a postdoc in Europe. So I thank you for your attention, and I'm ready for questions.
Okay, Enrique, thank you very much for such a beautiful talk. And yes, actually, we are looking for the questions of the people. So I have a question. So I will take advantage as a host. And one of the key aspects I got surprised at is that under your manipulation of the calcium stores, I didn't see a significant difference on the endocytotic decay on your capacitance traces. So
yes.
I would like to ask if you think that on these amacrine cells, there is any effect of the modulation by the release of internal calcium stores on the endocytotic decay, because in other models, it seems to be related, like in the hippocampus or even chromatin cells. So what you have to say?
Yeah. Chromatin cells are really interesting behavior with fast endocytosis. So in this study, we haven't looked at the endocytosis. Actually, endocytosis is very difficult to study because at least in the cells we're looking at, which were postnatal day 30, so fairly mature, but not really completely mature, there was quite significant rundown of the endocytosis. It occurs at the beginning after a whole cell, and then it runs down. In more mature cells, we saw better, more resistant, robust endocytosis. So, but we haven't explored yet if cyclic AMP affects that kinetics. It's something we're studying now very actively. We're very interested in the endocytosis, the mechanisms of these cells.
Okay, thanks.
Yeah.
So Leon is saying the results manipulating CMP and calcium signaling nicely show the potential for modulation of glycinergic inhibition. What do we know about presynaptic receptors on A2s that might exert such effect?
Yes. So what is the signaling? So dopamine is probably one of them that can affect cyclic AMP levels. And there's really nice papers by Erica Eggers in Arizona, and she shows with light stimulation that the glycine is actually inhibited with dopamine. And so if dopamine is increasing cyclic AMP, one mechanism that could potentially explain that is we also find in these cells that there's calcium activated potassium channels. And so if the stores are releasing calcium, they could activate these calcium activated potassium channels, and that could inhibit the release. So there could be very interesting calcium modulation that inhibits release, but also enhances release, depending on how your neuromodulator is acting. It could be completely different at night, as you know, with melatonin and other things at night. I think there could be quite a bit of plasticity. Maybe not as dramatic as yours, Jose, where the finch go blind, right? At
more or less
at 1 or 2 at night, and then their ribbons kind of disappear, and then they reappear at 6 in the morning. Maybe not that drastic in the mouse, but probably something similar, since dopamine levels change with light. Okay. It could be something completely different from dopamine that's actually elevating the glycine. Probably multiple modulations, yeah.
Mm-hmm. So Tom is asking, the calcium videos seem to suggest highly synchronized activity across the whole A2, which perhaps hints that the main driver for calcium is voltage. No? Do you see synapse flickering independently?
Yes, so that's a very good question. It could very well be that there is a depolarization, and that then causes calcium influx, which then activates the stores. So for example, ryanodine receptors are activated by calcium going inside the cell. So, and in that imaging, of course, we're not controlling the membrane voltage, so we don't know. So those are future experiments that we need to do, is actually do current clamp, for example, while we're putting those drugs, and see if there are voltage dependent changes. If it's all just internal and the membrane potential doesn't change, or are there voltage dependent changes? Completely agree with Tom. There's probably a bit of both, since there's probably global and local changes. Yeah.
Okay, so Chad Grabner is asking if can the calcium stores directly support release of glycine? And nice talk.
Sorry?
Sorry. If can the calcium stores directly support the release of glycine?
Yeah, so that's an interesting experiment. So we could have, for example, blocked the calcium channels. It'd be a little bit artificial, but it would be an interesting biophysical experiment. We could block the calcium channels and then put our EPAC activator, release calcium, and see if that's enough. My bet is maybe not too much, because we could block it with the 10 EGTA, indicating that the stores are a little bit far away, and so blocking with the calcium channels might affect that. But that's a good idea. That's an interesting experiment.
Okay.
Yeah.
Okay. Yeah. At the moment, there is not more question in the chat. I have another question, but maybe it's a technical question.
Yeah.
Because I think I missed the value, but you mentioned the size of single vesicles, glycinergic single vesicles. Do you have any idea about the size of glutamatergic vesicles of bipolar cells and how they relate? If they are, so if the size of glutamate vesicles are bigger on bipolar cells, or amacrine, I don't know, it's just,
yeah, that's a very good question. You know, there's a paper I remember from Tom Rees where he compared with EM the size of GABAergic, glycinergic vesicles, maybe even, and glutamatergic. You know, glutamatergic are somewhere around, about 35, 40 nanometers. And they're very round. GABAergic vesicles in EM tend to be a little bit flattened, might be an artifact of the fixation. With rapid freezing, they tend to be more spherical, but they're about the same size. They're pretty uniform, which maybe comes from the clathrin coat, you know. The size of the vesicle is kind of dictated by the clathrin coat. So if you think the vesicles are budding off, for example, from endosomes after the endocytosis, that, or maybe directly from the plasma membrane, they come already with kind of a uniform size. So I think they're pretty similar, pretty much around this value of 40 attofarads.
Okay.
Yeah.
We have another question. Simon Laughlin is asking is there any evidence that the synapses that act presynaptically within the synaptic complex are regulating vesicle release by their postsynaptic partners?
Yeah, that's an interesting question. So I guess, Simon, you're talking about retrograde messengers, maybe, right? If I understand that correctly, whether the postsynaptic partner could signal retrogradely. We have not seen like retrograde synapse that way. One interesting thing we did publish a few years ago in the goldfish retina, where we did paired recordings, was that if we stimulate very strongly in the goldfish retina, the MB bipolar cell terminals, those big terminals that I was recording from with Gary Matthews, we see that in about 30% of the cells, there's a really interesting potentiation of the AMPA current. So the glutamate does not change. You can just stimulate that repeatedly and it doesn't change. The capacitance doesn't change over a period of 10, 15 minutes. But the EPSCs get bigger and bigger and bigger in a percentage of amacrine cells. And we figured out that the mechanism was calcium permeable AMPA receptors. And we think that there are postsynaptically an incorporation of more AMPA receptors, maybe a phosphorylation of the AMPA receptors that is causing a maybe a more long-term plasticity in the retina. So it's kind of a taboo in the retina that to talk about LTP, you know, and LTD, but there could be post-tetanic potentiation. There could be, you know, after effects when you go into dark room and you stay for a very long time in there and you go back to the light, you know, it takes you several minutes to readjust. So the retina could have after effects that take many, many minutes to readapt and could involve some postsynaptic plasticity and some retrograde messengers. Yeah, that's a great question, Simon. We don't know what the retrograde messengers are. We know that there's CB1, there's endocannabinoids in the retina. There's pretty much any kind of, you know, there's NO that might be regulating the vessels. There's a lot of retrograde messengers in the retina.
Okay. So we have another question. So any roles for PMCA and mitochondria in calcium handling?
Yeah, that's a really good question. We did not look at mitochondria. Okay. And that's something we looked at in hair cells. And we're very interested in their energetics. You would think that the A2, it does have quite a bit of mitochondria with all this activity, this tonic inhibition that it's giving to set the resting memory potential of the retinal ganglion cells, the off-alpha retinal ganglion cells, that it's spending a lot of energy with all this inhibition that it's continuously sending to tone that resting memory potential of the ganglion cell. So mitochondria are probably very important. They are a sink of calcium. So when there's calcium around, they ramp up their ATP production. So they want to sense calcium, of course. But we haven't studied them per se.
Okay. Okay, so I think we are finished with the questions of the live chat. We already post the Zoom link if you want to join us for the discussion now in more detail with Henrique. So we already post the Zoom link, so feel free to join us. And thanks, Henrique, for accepting our invitation. It was a pleasure. And yes,
it was a pleasure. So do I just stay on, Jose?
Yeah, you just stay on. So
okay.
we will end the streaming in a couple of minutes. So we will give time to people for joining us. Oh, last question.
Yeah.
So can synaptic input from rod bipolar cells versus gap junction input from cone bipolar cells lead to different outcomes in calcium influx and glycine release?
Yes, certainly. One thing that we didn't show that we actually showed in a paper where we were looking at that plasticity from the calcium purple AMPA receptors is if you just puff AMPA onto the lobules of these A2 amacrine cells, it's really interesting. You get calcium rises. So we think that the lobules have calcium permeable AMPA receptors also. Not only their boreodendrites, as you know, Chavez and Jeff showed, but also the lobules. So even if you block calcium channels and MDA and just puff AMPA, you get these calcium permeable AMPA receptors, and it could be that calcium influx is enough to cause some glycine release. And that's input that's coming from the off bipolar cells. So it could be that they are depolarizing and directly, causing calcium influx that causes the glycine, which is a very different mechanism from what the rod bipolar cell does, which was AMPA receptors that go from the distal arbor and dendrites that need to then depolarize the whole cell until there's a release of glycine. So there could be a differential modulation in that way from rod bipolar cells, which are on type, and the off bipolar cells that are connected to the lobules, also via calcium permeable receptors. So it's interesting. The calcium permeable receptors appear a lot in the mammalian retina, but they're a feature more of very young, developing synapses in the brain, but they remain in the adult retina. So it's kind of an interesting thing, maybe not explored enough, and a source of plasticity, I think, for the retina also.
Okay. Okay, so thanks everyone for attending. So we will move now to the Zoom discussion, and okay, and see you next week. Next time.
Should I stop sharing?
Yes, okay.
Unless maybe I can stay here and I don't know. Can you guys see these slides? Yeah. Yeah.
I think we can.
Yes,
yeah.
I can.
This is a beautiful paper. I don't know if you guys saw, from Espen Hartveit and Meg Faruqi and Zunt, where they do triple recordings also on the A2s. And they show how, depolarizing the A2, you get these nice phasic glycinergic currents. Really amazing work.
So it's very nice to see you, Henrique, after a long time.
Yes.
And you actually answered a question that I was too stupid to think of. So my question was about these very direct synaptic contacts that you have in the complexes, where you might, for example, have an amacrine cell that synapses onto the terminal, the release, very close to the release site of a bipolar cell that's driving a ganglion cell. So what you have there is the opportunity to sort of micro-miniaturize very fast and simple processing. I say simple because there's not going to be very many other inputs coming in down the little dendrites that are feeding them. And it would be really nice to know how effective those complexes are at processing information.
Yeah.
But of course, it's an incredible technical challenge. I realize that.
Yeah. Simon, now I've been starting to try to learn a little bit of computational neuroscience and how to use Neuron and Python, and trying to get people in my lab to also get enthusiastic about that, because we know the morphology of many of these cells. We know a lot of the ion channels that are there. So maybe trying to get really good, realistic